Okay, perfect. Thank you very much. Hello, everyone. I'm Alberto Garcia, a teacher from the Polytechnic Master of Madrid of the Master of Quantum Technologies. And here we have to Elica, that is a quantum computational scientist from Boehringer. I hope I have pronounced it properly as well. So yeah, just without any further introduction, thank you very, very much, Elica, for being here. And this is all up to you. Thank you for being here.
Thank you very much, Alberto. I hope you see my screen.
Oh, yes, we can see your screen perfectly.
Okay. Thank you very much for having me, and also for the great opportunity to speak today. And I'm very excited, and I'm very passionate about my work about quantum computing use cases for the pharma industry. And I try to make my presentation today more targeted towards students, and it has also some elements of drug discovery as a whole, and then how we are hoping to change and improve on this process with quantum computing. So my name is Elica Kyoseva, and I joined BI about a year and a half ago when we set up the quantum computing lab of BI, but you'll hear about it more later. So initially, I would like to start by showing you and telling you what is BI. Many people are not familiar with our company, even though it's a very big company. It is also an old company. It was founded in 1885, and it is the biggest private pharmaceutical company in the world. And it is family owned. So it is pretty amazing that this is a 140 almost year-old business that belongs to the same family. And our revenue, of course, varies, but it's usually about 20 billion euros per year. And the focus of Boehringer Ingelheim is health of animals and humans. So we are both in animal and human pharma. And we are actually a huge company. We have more than 50,000 employees around the world in over 120 countries around the world. And we are very proud when it comes to quantum computing that Boehringer Ingelheim is the first pharmaceutical company worldwide that joined forces with Google Quantum AI for quantum computing applications and use cases. So in fact, drug discovery is a very computationally intensive field. And all of our drugs and all of the pharmaceuticals that we use are always based on a lot of calculations, really HPC, very heavy calculations in order to discover the drugs and also to make sure that they are safe. So Boehringer Ingelheim has a long-standing experience of using computing for research. And it was always a very progressive company. So this ranges from having a cutting-edge data collection process where we are part of many research projects. So we have over 500 research projects, many data points. You can see here about 380 million data points and many compounds, over 3 million possible compounds. And then in the last 20 years, let's say, we turned also towards AI and machine learning. And for that, we developed a machine learning tool called Adam that is helping us to predict the properties of the drugs and of the compounds, the possible drugs. And now, as a next generation technology, BI is actually very interested and very actively investing in quantum computing for drug discovery. And our lab was founded in 2020. Sorry, I don't know why it went back. So for the people that are not really familiar with drug discovery, I want to show you this pipeline. And as I mentioned in the beginning, drug discovery is a very computationally intensive task. So this means that there are many calculations that are taken over the process of designing and really having one pharmaceutical released on the market. And these calculations are done also over a very long time. And it costs a lot of money, of course. So the way that it usually works is we have a disease. So the first thing that we have to do when we want to treat this disease is to do target identification and validation. What this means is that we need to identify the protein in our body that is responsible for this disease. So in order to create something that can treat it, first we need to find out what is broken and what is not working. So this is the first step. And once we have the target identification, then we have a large amount of compounds. You can see even over millions of compounds that are possible candidates for a drug for this target. So then we need to go down this pipeline and to reduce the number of compounds that go from each and every stage to the next. So the second one is we do compound screening. So from the first few million compounds, we let about 1 million pass to phase 3, which is the hit validation. So these are already compounds that we know will be active and will be able to bind to the protein in interest, to our target, but we want to make sure that they are safe, that they're not toxic. We want to know how they will be absorbed in the body, how they will be also excreted from the body. So it is a very, very long process, even before it reaches the trials, the clinical trials. And of course, first we do it with cells, then you move on to animals, and then at the end is the clinical trials with patients. So you start from something that is about a few million compounds, and at the end of this computational process, you end up with one, hopefully, successful, safe drug. The time that it takes to go from the beginning of this pipeline until the end is about 10 years, and it costs more than $1 billion or euros investment. And the calculations that are involved are from many disciplines. So these are chemistry. Of course, because we deal with molecules, we deal with atoms, we deal with electrons, with their interactions. So quantum chemistry is a very, very big part of drug discovery. We deal with biological modeling. Of course, we need to find out how these chemical compounds are connecting to our cells. And as I mentioned before, to the whole body. We need to make sure that they also leave the body after they have acted the way that we wanted. Then we also have imaging. We want to be able, and we need to be able to see where the compound is distributed in the body. We don't want it to end up in an organ, or for example, in the brain that it is not intended to. So there are a big part of also drug discovery having precise imaging. Then we have material science. Material science as a field that is very involved in drug discovery, production, optimization of supply chain, and many, many other fields. So because of that, Boehringer Ingelheim really believes that quantum computing can be a real game changer for the field. And we hope that we will be able to speed up this calculation process and to reduce the time of over 10 years for calculations, and the same for the investment. Hopefully, that with quantum computing, we can make this process more efficient. And why can't we do it now? Why do we need quantum computers to improve this process? Why can't we do it just with our classical supercomputers? And the reason for that is that we all know that many problems become very hard, and they become very hard quickly. So suppose that we have all the data and models required for drug development, then it is still not enough to create the best and perfect drugs, because we need more. The process is very similar to searching for a needle in a haystack. And with our classical computers, as I mentioned, for this pipeline of going from 10 million compounds or 3 million compounds to just one final product, in order to do that, we really need to have the calculations to be very efficient and also very accurate. And this is really not possible with the current classical computing architectures. So we really feel that quantum computing can be a game changer. And unless this is possible, our drug discovery process and systems will stay the way that they are now. and we will never hit this exponential advantage that we require in order to make the process better. And the way we see that within Boehringer Ingelheim is kind of more or less in two stages. So in the first stage, which we consider the next 10 years, we hope that quantum computing will improve the process of today. So it will improve the efficiency and also the efficacy of today's process. The way that this will be resulted to our patients will be that we'll have more targeted drugs and also with fewer side effects. This will also have very good and important consequence on genome sequencing, where we'll be able to do faster and more accurate sequencing. And also for biomarker discovery, we need to find these disease-related mutations with better accuracy. So this is the first stage when we will be improving on the current processes. But the second stage, is where we will be also already disrupting today's process to identify the potential cures. So this means that maybe we will be able to overcome some parts of the computational process for the drugs, and we'll be able to simulate a big part of these calculations that I spoke about directly on the quantum chip. Of course, our ultimate goal, which I don't know if it will happen, would be to also overcome completely, let's say, the clinical trials. If you can model the whole body on a quantum computer with all their processes, with all its processes and biological systems. But if you could create a digital twin of the human body on a quantum computer, then we would be able to also overcome the clinical trials. I'm not sure that we'll be able to go that far, but this is kind of the ultimate goal. And because of that, and because of our belief in quantum computing for pharma, Boehringer Ingelheim actually set up a quantum computing lab at BI. And we are 5 scientists from quantum computing background that started working on applications of quantum computing to drug discovery. This is a photo of us taken last November in Vienna. And what is really interesting, and also for all of the students that are dialing in today and listening to this talk, was that we were set up as a completely remote team because we were hired during the corona epidemic when it was at its highest peak. So we were all based in different places, and we were also hired like that. Ultimately, we are kind of a bit converging. Everyone lives in a different city and almost different country. And when we took this photo in Vienna, this was the first time we actually met. After working for over a year together, it was the first time the whole team got together. So we are very much an example of a team that was set up during times when you couldn't travel and everything was done online, and how the borders really were opening. And this was kind of not the physical borders, but let's say the digital borders. And it became possible to work anywhere from the world. I'm personally based in Israel, though I'm moving to Boston in a few months. From top to bottom, this is Matthias. He's with quantum chemistry background. Then we have Nikolai Mo, who comes from IBM. He was to work there for many years, and he has also experience. He's a theoretical physicist and also has experience on superconducting circuits. My background was in quantum control and quantum optics. Mostly quantum engineering, kind of working towards the lower part of the computing stack. Then we have Mikhail Streif, who was just finishing his PhD with Volkswagen, working on QAOA. And at the end, we have Raffaele Santagati, who was an experimental physicist coming from Bristol, working with photonics circuits. And what was super important for the success of the team was that we worked internally in BI with the with the teams that were already very much involved in drug development, and this was their bread and butter. So we have computational chemists that really help us with the team, biologists, IT people, etc. So they really helped us in our ideation process, and even now. But because our goal was to actually find what doesn't work for BI with the current classical computations, our goal was to find out what first doesn't work, and then we had to think about ways to improve it with quantum computers. Okay, so now to go a little bit more into details, what we are mostly interested for, and even though I said that there are many disciplines that are involved in the discovery of pharmaceuticals, our work and what our team just decided to do was really to focus mostly on quantum chemistry. And the biggest thing about quantum chemistry is the electronic structure problem and the electronic structure Hamiltonian. Basically, this is the many body problem of interacting particles, and this is a very, very hard problem to solve classically, and the biggest supercomputers are able to do that for about, let's say, 20 particles, so 20 electrons, or if you use some really GPU accelerated systems, then maybe you are able to push this to 35 electrons. But this is where the current capabilities of classical computers are. And as all of you probably know, that to improve the capabilities, you need to double the size of the computer. So we don't expect that this will change dramatically. I mean, it will never be able to simulate the big enough molecules that we are considering for our drugs. So this is the electronic molecular Hamiltonian. We have different terms. We have the kinetic term, the potential term, and also the electronic repulsion term. As I said, it's many body interacting systems. So because of that, it is very hard for classical computers. And what is the ultimate goal and why we are interested in the electronic structure problem is because we want to solve the Schrödinger equation with this Hamiltonian, and we want to to find out at the end the energy of the molecule. So all of us know that Turing can be a game changer for this kind of problems, and I will show later why. This is that. Okay. And the reason for that is because encoding the electrons from the electronic structure problem on a quantum computer has its advantages compared to representing the electrons on classical systems, on classical computers. Because molecules are quantum systems, their mapping onto a quantum computer, which is another quantum system, is efficient. So for example, if we look at this graph here on my screen, if we start from electrons, then they have their possible orbitals, right? Here you can recognize the orbitals. Then when we want to represent these electrons on a classical computer, we need to go into a matrix, on a matrix description of the system. So in the case of n possible orbitals, then we need to have a matrix that is with dimensions 2 to the power of n times 2 to the power of n. But if we want to describe the same n orbitals on a quantum computer, we can do that only with n qubits. So you know, 2 to the power of n actually grows very, very fast. So because of that, once we have a little bit of a bigger system, let's say 50 orbitals or something like that, we cannot anymore do that, even less, maybe 30. We cannot do that anymore on a classical computer. But we would require only about 30 qubits in order to do that. And for example, if we look at the oxygen atom, and we consider 10 orbitals for its description of the electronic wave functions, then classically this has to be represented with a matrix that is 1024 times 1024 matrix size. You know, 1024 rows with rows with 1024 columns, while quantumly we can represent this only as 10 qubits. So you can see the huge advantage of using quantum computers for describing quantum systems. And this comes really from being able to represent the system in a much more efficient way than when we are trying to represent it with the classical computers. And why are we interested in such big systems? Well, here is an example of the molecule part of it. This is the example of the Jardiance molecule, which is the best-selling drug of BI. And this molecule has, I think it was about 230 something electrons. So as I mentioned, the classical computers power finishes at about 30 possible orbitals for these electrons, while here we have many more electrons, and then the orbitals that we need to consider for them are also many more. So we will never be able, with classical means, we will never be able to represent this molecule exactly on a computer, because if we have the number of particles that we want to describe, if it is about 200 electrons, then the bits, the classical bits that are required for their encoding are 2 to the power of 200, which is approximately 10 to the power of 60, which is more than the atoms in the planet Earth. So we'll never have such big supercomputer. And maybe you're wondering, okay, so how did the BI make all these calculations if it was not possible with the classical means? And the way that people do it now, including BI and everyone else, is by doing a lot of approximations. So the way that industry is working around this computational exponential bottleneck is by removing some of the interactions, because we say, okay, not removing, but approximating them to zero, and employing a lot of approximations, and they work. Obviously, that's why this drug is good and it's selling and it's helping our patients, because these approximations work. However, if we want to have a perfectly exact representation of the molecules and of their interactions on a quantum computer, such that our calculations of their properties will be really exact, then we are not able to do it with classical computers. So can we do it with quantum computers? I'm sure all of you probably have already seen this graph from Google. We are still also not able to do these calculations with quantum computers. However, we are kind of in the dawn of the NISQ era. And here on this graph, the red dot represents Google's quantum supremacy experiment. And it is still not in the NISQ in the sense of useful quantum algorithms. However, it is in the supremacy regime, where classical computers cannot compete anymore with the quantum computer. But ultimately, to have something very useful, we would need to go to the error-corrected quantum computers, because even for NISQ, still the error rates are sizable. They are not something that we can neglect, and they do limit the depth of the circuits that can be executed. So because of that, in order to really get the benefits from quantum computers, we really need to go to the green area on the graph and have error-corrected chips. We have some predictions that this will happen at about 1 million qubits or more, and there are companies that are promising, of course, chips with a million or so physical qubits. So we are really hoping that in the next several years we will see error-corrected qubits and also 2-qubit gates, etc. But it's important to note that, in fact, last year we did see the first experiments and realizations of error-corrected qubits. So what are the current advances, and what needs to happen is that the companies should really start investing now. And something that people, I think, don't really realize until they start working on these topics is that we can't just ask for the quantum hardware to become better and to have, error corrected even device. We also need to have computational improvement on the algorithmic side, because if people sit down and do the calculations and find out how many actual logical qubits we need for the algorithms that everyone knows, and how much runtime it will take on an error corrected device, then you see that this is actually still prohibitively large. And it will not be possible, even if tomorrow we have, let's say, 1,000 qubit error corrected device, we still won't be able to run Shor's algorithm on it, because it will take too long. It will take 100 or so years to run like a complicated algorithm. So we definitely need also to work on the algorithmic side of things, and the development of software and hardware has to happen at the same time. And in that sense, this is a graph from McKinsey, and it's really for the quantum, not for the quantum, it's really for the drug discovery industry. So for the stage 1, which is in the next 5 years, and the stage 2 expectations, which is in the next over 10 years from now, we are kind of already after the incubation period. Many pharmaceutical companies are already involved in some way in quantum computing. Most of them are just keeping an open eye. They're participating in consortia, or they're attending, conferences. Maybe they have a partnership with a few companies from outside to kind of understand in which part of their product pipeline quantum computing can help. But in the next few years, now we are kind of entering stage 2, where we have, and we will start to see NISQ devices that are already in the quantum supremacy regime, and we have to find smart ways to use them in order to have some benefit for our companies. And then in the stage 3, which will be probably after 2030, we will have quantum error corrected devices, but also by then we need to improve the algorithms. As I mentioned earlier, we need to make them more efficient with better scaling, so that the time it takes to run them on their corrected devices is still not prohibitively large. And now I will go into the more technical part of this talk. The first part was really kind of an overview of pharma and quantum computing to the pharma industry. And this one is our first paper that came out from our quantum computing lab. And it is about calculating interaction energy with the quantum computers. And the focus of this paper were actually NISQ quantum computers. And we did it in collaboration with our partners from QCware. We are proud because it was just accepted in chemical science, and it will be also on the cover of chemical science, I believe, next month. So now we need to go a little bit deeper into details. So what are interaction energies? Basically, it is what we have an idea of from our studies of physics. But when we think about it in the concept of drug discovery, basically the interaction energies tell us how well does a ligand bind to a protein. In the beginning of my talk, I talked about target identification. So this is our protein, the one that we want to treat because it is causing a disease in our body. So we want to bind our active molecule to it, so that they can interact, and they can and the ligand can actually change the behavior of the protein. So why is it important? It is very important. The interaction energy calculation is very important because it directly determines the efficacy and dose of the drug that we want to give to the body. It's always important to remember that we always want to give low dose pharmaceuticals and drugs, because they always have some side effects. So you always want to give the least dose possible, so that you would minimize the occurrence and likelihood of side effects. So there are two ways to calculate, maybe there are more, but in this talk, I'll talk about the main two ways to calculate interaction energies. One of them is the naive supermolecular approach, which is on the left, which means that we do subtraction of the ground state energies. So if you think, if you look at my figure on the top where I have monomer 1, monomer 2, so they bind together and they form a dimer. So what it means is that in order for me to find out what is the interaction energy between monomer 1 and monomer 2, I need to calculate the ground state energy of the joint system, which is the dimer. And from it, I need to subtract the energies, the ground state energies of the individual monomers, 1 and 2. Here in my labels, it's A and B, but it should be 1 and 2 to correspond to the graph. So this is the so-called supermolecular approach. You take the whole system, the energy of it, and from it you subtract the energies of the subparts of the system. However, this approach, when taken in the concept of drug discovery, has some issues. So one of them is the basis set superposition error, which comes when you need to take into account the basis set of the individual monomers. And the other error is actually that it requires, this approach, an enormous precision. The reason for that is that the energy of the monomers and also of the dimer are usually on the order of 1000 kcal per mole, while the interaction energy is much smaller, it's much weaker, and it's only usually around a few kcal per mole, maybe 5. So what happens is that from one big number, you need to subtract other big numbers, and you need to do this with a very high precision in order to be very accurate for the difference between the energies. So this requires an enormous precision, and it is in fact very difficult to achieve, especially on quantum computers, because this kind of a high precision would require very, very deep circuits. So we want to do something that doesn't require such a high precision in order to have shorter circuits. And this is what we did with QCT21 for this paper. We used something called the Symmetry Adapted Perturbation Theory, or SAP. And what it does is it overcomes this subtraction problem, and it directly computes the interaction energy of the two monomers as a perturbation series of their particular type of interaction, intermonomer interaction. So we have that the total interaction energy is equal to electrostatic term, exchange term, and for them we have their exact representation. We have then second order terms, which are dispersion and induction, and so on. So you can continue, because it's a perturbation theory, you can actually continue with more and more terms, but it's usually enough for people to calculate the first order terms, the electrostatic and exchange term of the energy, and the second order terms, which are the dispersion and induction terms. And as I mentioned, we were interested to do the symmetry adapted perturbation theory in NISQ. So for that, what we did was we considered, of course, the variational quantum eigensolver, because this is the NISQ algorithm for quantum chemistry. And what we did, and of course, also ultimately it was a hybrid algorithm. And in our paper, we show the calculation of the first order terms of the energy with a hybrid quantum classical approach. So what we did was that for the first order terms, we need only the ground state wave function of the individual monomers. So these are monomers A and B. And what we assumed was that, and also what we implemented, was that the ground state wave functions of the monomers are actually given by a variational quantum eigensolver. So in here, my equations for the energy, for my expressions for the energy of Ca and Psi b are the ground state wave functions of the monomers A and B, and they are in fact generated on a NISQ quantum computer using the variational quantum eigensolver. In our first part of the paper, in this paper that is now getting published, we actually did this for the first order terms, and the second order terms are following. So today I will show you the results only for the polarization, the first order polarization and exchange terms. So even with quantum computers, we need to do some approximations in order to be able to fit these systems, these monomers, on a quantum computer. So the first approximation that we did was we used the active space approach, which means that you have some core electrons that you do not you consider them frozen core. Then you have the active space, and then you have virtual orbitals. So by using this active space approach, what you basically do is you reduce the number of orbitals that you need in order to describe your system. And after that, in this active space approach, we found the ground states of the monomers by using VQE. And then what we did was basically from the VQE, once we have generated the ground state wave function of the monomers, we measure them. We measure the ground state wave functions, and generated from them the 1 and 2 body density matrices, which are the gammas here in my graph. And then we used classical post-processing, the classical SAPT approach, in order to calculate the interaction energies. So the way that this process works is you start with the protein, then you do the active space selection. Then in this active space, after the active space selection, we do VQE in order to generate the ground state wave function. Then we do measurements, and with the measurements, we extract the 1 and 2 body reduced density matrices of the wave function. And then we do classical post-processing in order to rate at the end which drugs or which candidate molecules are best binding to the target protein. So this is how our wave function looks like. This is the simplest possible one, let's say the shortest depth wave function circuit. And for that, to generate the wave function for the monomers, we use the unitary cluster Jastrow wave function, where the parameter K actually tells us how deep is the circuit, where K equal to 1 is the shortest possible ansatz. And here in my graph of this circuit, we showed the VQE circuit for K equal 1 in the case of 4 spatial orbitals or 8 qubits in the Jordan-Wigner representation. So this is how the smallest circuit looks like. And remember that here this G are Givens rotations, and PX are also, they're like 2 body operators. So they're not individual, let's say, single qubit gates, they're multi-qubit gates. So we can, of course, write down even in more details in terms of single and 2 qubit gates, but then it becomes too complicated to represent. And this is the depth for our shortest possible VQE wave function. Of course, when we improve and we increase the depth of the unitary cluster Jastrow wave function, then we get more accurate wave function, and we will have a better calculation for the SAPT. And the other thing that is worth mentioning is that the number of measurements that we require to do it is usually, in order to get the 2 body reduced density matrices, is usually on the order of n active space to the power of 4. So the number of orbitals to the power of 4. And now that we had derived our calculations, so what we did was we applied, of course, this process to a system, and this system is the benzene p-benzyne dimer, for which we know that it is actually difficult to calculate, because the p-benzyne, which is the one at the bottom, the ring at the bottom, it has a biradical ground state. And this means that it also benefits, it needs to be described with a multi-reference approach. What it means is, it means it's difficult for classical computers. So it is hard to describe the state of this p-benzyne on classical computers. So that's why we chose it for our quantum computing experiment, numerical experiment. So what we did is for this system, we actually calculated the error in the total energy and also on the individual components of the interaction energy. So this is the electrostatic and the exchange for different depths of our VQE ansatz, which is K, which is this factor K, which ranges from 1 to 5. And we find that actually the something I didn't mention before, but there is something, there is this kind of figure of accuracy in quantum chemistry, which is called chemical accuracy. So in order to be within chemical accuracy, this is usually around 10 to the minus 1 kcal per mole. So whenever your calculations are within this accuracy, then you are happy. And we found here that our calculations were actually within chemical accuracy, even for the shorter depth VQE ansatz, which was in fact very, very promising to us. And then what we did was basically we changed the distance between the p-benzyne and the benzene ring. And we compared the error in the electrostatic energy as a function of the distance. And we also compared it to classical calculations done with the restricted Hartree-Fock methods. So the orange and the green are the wave functions and the results generated with wave functions from a quantum computer, while the blue line on top is done with the classical calculations of RHF. And here you can see very clearly that when we use the wave functions generated by quantum computers, we get a much better behavior and scaling of the error. So this was, again, very promising for us. And the last figure is we did the same for the other term, for the exchange, for the first order exchange term. And again, we compared the quantum computer generated wave functions to the classical calculations, and we found a better behavior and less error in these terms compared to the classical wave functions. And the reason for that is because, as I mentioned in the beginning, one of these systems is actually difficult to calculate with classical means. So then when we do approximations, they are not very accurate. And because of that, when we avoid these approximations by using a wave function generator from a quantum computer, we get better and more accurate results. After this calculation, we also did one more, which was when we looked at real proteins, and the protein that we chose is the lysine specific KDM5A protein. And the reason that this protein is difficult to describe with classical methods and classical computers is that it has an iron atom inside. So this makes it more difficult to describe. So then also the total region, then if we look at details of this protein, the total region around the active space, this is the place where the protein will bind to our drug of interest, has about 380 atoms. So it is a big protein. Proteins are big. So by using one particular basis of quantum chemistry, we ended up with 1,482 electrons and 2,214 orbitals. So you can see that this is way too many to represent on a classical computer. So after that, what we needed to do is we needed to reduce this active site in like active space representation by using this AVAS technique. And at the end of it, we ended up with 36 electrons and 27 orbitals. And then we needed to even reduce it more because even that is still too big for classical computers, but also for the current NISQ devices. So at the end, we ended up with having 8 electrons and 8 orbitals for our calculations. So because this method is to calculate interaction energies, as I mentioned, this tells us how a drug binds to a protein. And as in any case for any energy calculations, we are not interested in absolute values, but we are interested in energy differences. So what we ended up is we basically ended up comparing how these 5 molecules that are on the left side of my screen bind to the target protein, and we needed to rank them. So we need to say which one will bind the strongest and which one would bind the least. And again, we did the calculations with comparing with classical methods. So this is the restricted Hartree-Fock with also DFT, and also with our method of SAPT. And so I don't bore you too much with the details of these graphs, but what we found out is that calculating and doing these interaction energy calculations with restricted Hartree-Fock is insufficient, and we get some errors. For example, here you can see that compound 4 on the top graph, compound 4, let's see. Oh, okay. So we can see that compound 4 is really, its energy is really miscalculated by using restricted Hartree-Fock. And then with some DFT, it gets improved, while our SAPT second order approach is actually calculating all of these interaction energies properly. And when we do that for the full configuration interaction, so this is this CAS-CI, we find that there is almost no difference between the results that we get with SAPT VQE and also SAPT CAS-CI. Okay. So this is my this was my last slide, and I'll be happy to answer questions. But what are the conclusions and the takeaways that I wanted to give to you is that we need to be very smart when we select our use cases of quantum computing for the pharma industry. The reason for that is that it does not make sense to solve with a quantum computer something that will be easily solved with a classical computer. I know this sounds funny, but actually it is not that trivial to find which systems are difficult to calculate with a quantum computer when we consider real molecules and real protein, which most of them don't have a lot of correlations inside. So most of them are in fact described relatively well with classical means and approximations. So when you want to do a use case for quantum computing, you need to find a use case that is indeed hard to do on a classical computer. In order to see these improvements, we need a substantial development on both software and hardware sides of the stack. And this will be needed in order to make quantum computing useful. Having only quantum hardware will not be enough. And the last thing is that I do believe that companies should start investing in quantum computing now in order to not be left behind and to be, to really lose using quantum computers for their use cases. Because the system is currently, and the whole industry is still pre-competitive. It is still very much research. Most of the results are, but probably this will change soon. And then companies will start to keep secret of their discoveries and the applications of quantum computing. So it is important to start investing now. And thank you very much for your attention. And I'm happy to take any questions if you have.
Hello, Elica. Thank you very, very much for that. It has been a very, very great presentation, really. In fact, I will see the seminar again because I think it's very, very interesting.
Thank you. Thank you, Will.
Is there any further question for Elica? Okay, so I think we have done because there is no further questions, Elica. So thank you very, very much for being here. It has been a great pleasure, and hopefully we will have you next year again.
Yeah, thank you and good luck to everyone.
Yeah, thank you very much. Bye-bye.
Bye-bye.